All right. Thank you so much for your patience with our technical difficulties. Let's start fresh. So good morning, everybody. My name is Dora Tortarolo. I am a computational biologist, and I work at the Department of Computer Science at the University of Torino. I am part of the quantitative biology lab led by Professor Cordero and Professor Beccuti. And today, my colleague Grete Francesca Privitera and I will give you an introduction into the world of microbiome, and we will provide you with some tools to analyze specifically 16S RNA data. So let's start off with understanding and defining the microbiome. So there are very different definitions of the microbiome, and they encompass different levels of biological organization. So from individual microbial genes and genomes to different species and strain composition to community structure and their functional capabilities. The simplest organization is that of the collection of genes and genomes. That is the metagenome. The collection of all microorganisms living in a specific environment is referred to as microbiota. The most complex definition is the one that involves both the microorganisms living in a specific environment and their distinct properties and functions and how they interact with the environment. And this is defined as the microbiome. So the microbiome involves all of the living microorganisms and the molecules that they have produced, including structural elements such as nucleic acids, proteins, lipids, and so on, and their metabolites, all of the signaling molecules that they produce to interact among themselves and with the host. Microbial organisms belong to 4 kingdoms: the bacteria, archaea, eukaryota, and viruses, and they are literally everywhere. They are ubiquitous, and they play crucial roles in almost all of biological processes in ecosystems, from natural environments such as soil, which has the largest microbial biomass on Earth, to the human body, on which I will focus on today. So in the human body, there are trillions of microbial cells, and each human body hosts thousands of microbial species at once. And they are crucial for all of our functions. So these include digestion, immune regulation, metabolism, and even neurological signaling. And their role is so great in development, growth, and health that really individual phenotypes are seen as the result of the combined expression of the genes of the hosts and the genes of the microbes. So if the human genome has about 20,000 genes, the host and the microbiome together has 33 million genes. So really, we can look at the human body as a holobiont. So the association of our body and its associated communities of microorganisms. And our bodies interact with microorganisms through wide range of different types of interactions, from mutualistic interactions to parasitic interactions. Interestingly, the microbiome is actually extremely heterogeneous. So unrelated individuals in the population have on average approximately 30% of species that are shared among their different guts. And if we look at strains, which are microorganisms that belong to the same species, but actually show different functional capabilities that they have acquired through horizontal gene transfer, for example, through plasmids or transposons. If we look at strains, then unrelated individuals only share less than 0.1% of strains. So really, the microbiome is a fingerprint that is almost unique to each individual. While there is a really high amount of inter-subject heterogeneity, even within a single host, so a single person, the variability is huge. So different anatomical sites really harbor distinct microbiomes. And this is due to, first of all, local physical-chemical environmental factors, for example, pH, oxygen tension, moisture, and nutrient variability really select for different taxa. There's host factors, for example, the tissue architecture or how much immune effectors can reach those compartments, and also how these organs relate to one another. So the biogeography of the organs influence the microbial composition. Organs that are contiguous one to the other, for example, the oral cavity, the esophagus, the stomach, and the gut and the intestine, are contiguous, they're connected to each other, and these organs show a gradient of shared taxa. Within these organs, we have even more variability, and that is due to the formation of different microhabitats that maybe can create different microbial compositions due, for example, to the structure of the organ, if they have a mucosal layer and a lumen within them. So how is this microbiome acquired? Microbes don't colonize the human body before birth in healthy pregnancies, since the human womb and human fetuses are considered to be sterile. But humans encounter microbial species as soon as they get out of the womb and they reach the environment. And the first event of acquisition is the delivery mode, which can be, a natural birth through the birth canal, or it can be through a cesarean. The second event is the feeding mode, which can come from breastfeeding or from bottles of formula. And of course, the human contact. And how this interaction occur really shapes the microbial composition of babies. As babies grow, they come into contact with the environment. And so the rest of the world becomes a source of colonization. Mothers and babies share about 50% of the gut microbiome up to when the baby is 1 year old, and this percentage drops to 25% by the second year of age. When they're 3 years old, toddlers really start to have a mature and stable gut microbiome, which is similar to that of adults. And interestingly, adults actually have many microorganisms that are not present in infant microbiomes. But very little transmission is occurring from the environment, since most gut microbes are strictly anaerobic, and so they would not survive out in the open. Most of our acquisition of the microbiome as adults comes from proximate individuals through horizontal transmission. So individuals that live in the same household share more gut microbiome than people that don't live together. Then up to 3% of our gut microbiome comes from foods, and especially from fermented foods. So we are familiar with some foods that are fermented, for example, bread and yogurt, of course, wine and beer and vinegar. But sometimes we forget really common ones that are also fermented, such as chocolate and coffee, and even vanilla. Finally, there has been some evidence that transmission can also overcome species barrier. So people that live with pets and stock breeds often find that they share some microbes with them. So an interesting example is Escherichia coli. Some strains of E. coli have been found to be more frequently shared among dogs and cats and humans that live together than between humans that live in different households. There are also artificial inducers of the microbiome, for example, through traditional probiotics or live biotherapeutics. And recently, also fecal microbial transplantation has arisen as a promising clinical procedure. In this procedure, there is a transfer of processed stools from a healthy donor to a patient's gut with the goal of correcting some imbalance in order to restore health. In fact, since microbes are involved in all of our physiological functions, when the balance between the microbes and the host is disrupted, we can encounter disease. Usually, this imbalance can be referred to dysbiosis, and it's characterized by a reduced diversity, the loss of beneficial taxa, which aren't able to prevent anymore the overgrowth of pathogenic and harmful microorganisms. This results in an altered metabolite production and also immune dysregulation, which can promote chronic inflammation and even autoimmunity. This can occur both locally within the organ where the dysbiosis is occurring, but it can expand throughout the body. For example, in the gut, it can cross the intestinal barrier, and metabolites and species can reach the entire body. Of course, we have some diseases that are well known to be influenced by the microbiome, for example, inflammatory bowel disease, which is often associated with the leaky gut syndrome. So this syndrome in which the gut lining becomes more permeable and microbes can reach the blood circulation and reach other organs. But there's also been other cases in which the role of the microbiome has been found. For example, heart disease, which is often correlated with a dysbiosis in the oral cavity, and several types of cancers. So alteration of the microbiome have been found in tumoral tissues and has been implicated in disease progression and response to treatment. So it's really crucial for us to understand the role of the microbiome in disease. And so to understand this, we must answer some questions. We must understand who is present, so which species are present in that tissue. What do they do? Which functions they might exert in that tissue, and how these functions may influence the host and its health. But actually characterizing the microbiome is very difficult. So traditionally, we would isolate microbes and culture them in order to characterize them by direct observation. Now we fully sequence their genomes in order to understand which genes they carry and what they might actually do. But it's really hard because less than 2% of microbes can be cultured in the lab. Furthermore, by isolating them and selecting them, we lose any information on the quantity of that bacterium or microbial species in that compartment. And also we lose insight into the ecological interactions of that microbe with the rest of the community. Conversely, it's possible to sample an environment and directly sequence all of the genomic content present in that environment without selecting first for specific species and culturing them. So metagenomic sequencing has several advantages, including being applicable to theoretically all of microbes present in a sample, and it's also high throughput. However, its main limitations are that it gives an output that is very compositional and complex and computationally demanding to analyze. Today, we will focus on metagenomic sequencing, and there are different approaches through which you can do that. There is amplicon sequencing and shotgun metagenomic sequencing. In both cases, you start from a mixed microbial community and you extract the DNA. In amplicon sequencing, once you've extracted the DNA, you only sequence target genes. And these target genes must be both universal and variable. The idea is that they act as taxonomic barcodes. So the conserved region is necessary so that all species belonging to a specific kingdom will be identified through that conserved region. For example, all bacterial species must have a portion of that gene that is present in all bacterial species. Then it also has to have a variable region, and that variable region allows us to discriminate among different genera or species. The most common targets are ribosomal RNA, which is very highly conserved among different species belonging to a kingdom, but also allows us to identify different species within that kingdom. And for bacteria and archaea, we target 16S RNA gene. For eukaryotes, we target 18S RNA gene. And for fungi, we target ITS1 gene. You can also target some functional genes that are specific for specific metabolic groups belonging to different kingdoms. So in amplicon sequencing, we extract all of the DNA, and then we perform targeted PCR against our gene of interest, for example, 16S. Once that gene has been amplified, it gets sequenced. And the sequencing output is then compared to a database that contains all of the possible, all of the known reference target genes. So all of the target genes that have been already identified for different species. If our sequenced target gene maps against a target gene in the database, we know that that target gene belongs to a specific genus or species. In shotgun metagenomic sequencing, instead, we analyzed the entire content of the DNA extracted from our sample. In this case, from our DNA extraction, we fragment the different DNA pieces into smaller fractions and then directly sequence these fractions. The reads that we obtained are again mapped against a reference database, which in this case doesn't contain only the target genes, but really it contains the entire genome or most of the genome of the known microbial species. So when enough reads map against a reference genome, we can assign the correct classification to that pool of genes, of reads. That way, we know which bacterial species or microbial species are present in our sample. So both techniques allow us to define which species are present in our sample. Taxonomic metagenomic sequencing also gives us an idea of the functional repertoire of the microbial species present in the sample, since we know which genes each species carries. If we want to know also what those species are actually doing in that samples, then we have to move to the metatranscriptomic technique. In this case, we're starting from a mixed microbial community, but we're extracting RNA, and the RNA sequences are then sequenced and mapped again against a reference genome. This way, we know both which species are present, which species are alive in that sample, and what they're actively doing in that moment. So which genes they are transcribing. All of these techniques come with advantages and limitations. So amplicon sequencing is the lowest in cost and doesn't have any problem with host contamination, since we're actively selecting only for microbial target genes. But it does have some limitations. The resolution is limited and it's biased by PCR and primers. Shotgun metagenomic sequencing instead has a higher taxonomic resolution, and it allows us to have information on the functional potential of the community. However, it's more expensive, and it's really limited by host contamination, since all of the DNA content in a sample is being sequenced, and we can remove the host DNA content only computationally. Finally, metatranscriptomic sequencing gives us information on population dynamics. So it allows us to understand how bacterial species or microbial species are growing and dying in our samples. And also it allows us to characterize what they're actively transcribing, so which genes are actively expressed in our sample. However, it's much more expensive. And the main limitation is that RNA is highly unstable. So we need a lot of microbial RNA for sequencing, and our samples must be processed immediately after collection. So today, my colleague Grete will tell you about how to analyze amplicon sequencing, and specifically 16S RNA data.
Hello everyone, I'm Grete Francesca Privitera from the Department of Clinical and Experimental Medicine of the University of Catania, and as my colleague Dora said before, I will talk about amplicon sequencing, called also metataxonomic. That is the way of sequencing the 16S ribosomal RNA gene of bacteria. Why this specific gene? Because it is a universal primer that binds virtually all prokaryotes and has also 9 prevariable regions from V1 to V9 that evolve fast enough to distinguish organisms both at genus and species level. In this picture here, we can see all the possible primer, so the prevariable region that we can choose, and the most used are the V3, V4, and V6, V8. Why? The V3, V4 because are the standard for Illumina MySeq clinical microbiome study. This because they offer an excellent balance between taxonomic resolution and amplicon size, because it's possible to sequence them in paired end with 250 pair-based and 300 pair-based. And the V6, V8 instead are usually used for rhizobiome, for plant microbiome in general, because they don't permit the sequencement of the chloroplast. This is a summary of how 16S amplicon sequencing process work. As Dora said before, we take the 16S RNA gene of the ribosome, and it is amplified in PCR. During this amplification, for each sample, it is assigned a barcode. The barcode is a short nucleotide sequencing that tag each sample. In this way, we will be able, when we do the downstream analysis, to recognize each sample thanks to this barcode that is a short sequencing of 8-12 base pair. This 16S gene is amplified sequencing in the MySeq usually, and we will have at the end the possibility to study the microbiome inside our sample, starting from the domain to the species at taxonomic level. But how we group the 16S sequencing into biological meaningful unit? We have two possibilities. We have the OTU and the ASB. The OTU, the operational taxonomic unit, are not very much used these days because they are sequences that are clustered together by a similarity threshold. The similarity threshold is 97% nucleotide identity. And how it works, the 16S RNA gene is sequenced. After that, all the sequences are clustered thanks to this threshold, and all the sequences that are inside this threshold are represented by a representative sequence. But this is a limitation because the clustering introduces arbitrariness because we have decided the threshold, because it's this threshold is the boundary between bacterial species, but it's a simplification of the biology behind them. And the sequencing errors are not distinguishable, and the results are not reproducible across studies because the clustering depends on the dataset used, on the unification of the clustering of this sequence. Instead, nowadays, the unit used are the ASB, the amplicon sequence variant, and they are used with tools like Dada2 and the Blur. We will talk about them in a minute. And they are not a cluster. They are a unique sequence. In fact, even if there is only a difference of a single nucleotide, we have two different ASB. In fact, the advantages are the single nucleotide resolution, because closely related strains can be distinguished, the reproducibility, because not doing a clustering, we have always the same sequence, and that we don't have to choose an arbitrary threshold. How a 16S pipeline work? Here there is a summary. We have, first of all, the CINI analysis. CINI is the tool standard, the gold standard tool used nowadays. We have at the beginning the raw reads that came out from our sequencement. The raw reads are trimmed to delete the adapter and the low quality reads. After that, the files are imported inside QIIME2 because QIIME2 only accepts artifacts in QZA format, so it is important to importing them. After that, we can do a quality control on our sample, and we can build the feature table. The feature table will be necessary to do the next analysis, that will be the phylogenetic analysis that will permit to us to calculate alpha and beta diversity, and the taxonomic analysis that will permit to us to recognize each bacteria. And after that, we can analyze with data stream analysis using R, the alpha and beta diversity statistic, understanding the significance, the diversity between groups, and the differential abundance analysis that measure the differential abundance between two groups, calculating a p-value. Another analysis that can be done using the feature table is the functional abundance analysis with the software PyRAS2. This software permits to understand which metabolite, which pathway are influenced by the bacteria, the microbiome that we found in our sample. And also in this case, it's possible to do with R a downstream analysis, calculating a p-value for the metabolic differential abundance analysis. We will see each step now. QIIME2 can be downloaded in this URL. It's very easy to install. It is possible to install 4 different environments. For example, there is also an environment for the shotgun analysis, but the distribution that we need for 6CNS analysis is the Amplicon one. To install it in Linux, we need to create a conda environment, but the user manual guide us through all the steps, and we can also copy and paste the command, that is this one, that change for every update of the software. The last update was in January. After that, as I said before, we have to import the file in QIIME2 because QIIME2 accepts only QIIME2 artifact. But from sequencing, we can have 3 types of data: file multiplex with barcode in FASTQ file multiplex with barcode in TXT file, and file that are not multiplexed. When the sequences are multiplexed, we will need to put in the same folder the FASTQ of the sequence, and the FASTQ contain the barcode that will be like this one. And we can use the QIIME tool import with this type here. I put as the sample the parent and one, but it is possible also to do this in single and changing only paired with single. And we will have the QZA file with our sample. If we have the barcode not in a separate FASTQ file, but in metadata, we have to put inside the same folder the FASTQ with the multiplexed file and the TXT. And the command is the same one, it's tool import, but we have to change the type with multiplexed paired and barcode in sequence, because the barcode are already in the sequence and not in another FASTQ. And also for this one, it is possible to change paired with single. At this point, regardless of the flow that we have used, we will need to demultiplex the file, and we will use this command, QIIME to demux amp. Here I put the single and the sample, but it can be changed with paired, and we will have a demux file at the end. And the third option, when we have sequences that are already demultiplexed, we have to create a manifest, that is this file here, where we have to put in the first column the sample name, and we have to create 2 other columns, one with the forward absolute file path and one with the reverse absolute file path that indicates to QIIME2 where to search the FASTQ. the command is always tool import, but we have to change the type with sample data, in this case paired-end, but can be single-end sequence with quality. And we will have again the demuxqza file. In all cases, the demuxqza file should be summarized in a qzv file, because this is a file that permits to us to visualize our result with Chain2View, that is a web server service by Chain2. When we put the file demuxqzv in the Chain2View website, we will obtain these plots for when we have paired end, only on one plot when we have single end. And this plot is the quality plot that will permit to us to cut the low quality sequence. Clicking into this box, it will appear this table under the quality plot that will say to us the position in which we are clicking, and the bottom of the box, the middle of the box, the top of the box with the quality score. Normally, in a microbiome 16S experiment, it is common to take only the sequence that have a threshold of 30, but sometimes it's difficult. So we can trim down to a quality of 20 to avoid losing too many base pairs. And here, for example, we can see that the sequence base from 0 to 35, more or less, have a very low quality, arriving to 10. So we can decide to cut, like here, more or less 35. And we have also to choose where to cut in the right part of the graph, the plot. And we can cut here at 210, more or less, because if we follow the 30 line, we will see that from here it drops. So we can stop here. We have to do the same thing in the reverse reads, but the cut does not need to be equal to the forward reads. The important thing is that we have at least 20, 30 base pair of difference between the forward and the reverse reads, because we need them to match. So here we can keep the first part, so we cut at 0. And we can decide to cut more or less right here, because after that, the sequence of the base drop. This is a second experiment. I put it here to show you that not all the quality plots are equal. Darker is the plot, more sequence there are in this, more reads there are in this point. Here, for example, the major part of the sequence have a 35 quality. How we can do this quality control cut? We use the data2 that we, as we said before, command, and we have to trim left for forward and reverse and right for forward and reverse. In case of single end, you only have to do one cut for left and one cut for the right. Data2 can be used also on R, and it can be installed through BioHack Manager, but I always suggest to use Chain2 because it has a lot of features that are easier to use, and also it's the gold standard. After this quality plot cut, we will have the feature table and the statistic of our cut. It is important to watch our statistics. Why? Because the cut, it's not always good at the first time that you do it. And you can understand if your cut is okay watching the percentage of input that passed the filter and the percentage of input merged. If these two parameters, this one and this one, more or less match, it means that the cut worked well. If you see, like here, 94%, and in the input percentage of input merged, like 5%, the cut has to be redone again. Unfortunately, the cut has not a fixed parameter. should be able to choose on your own. Now that we are sure that the cut went okay, we can inspect the feature table. The feature table can be inspected again with the QIIME2 view, and it will permit to us to understand how many reads we have for sample. For example, here in 30F sample, we have 945 reads. Why we do this inspection? Because it happens sometimes that some sample hasn't been sequenced well, and maybe here it can exist a sample called 1F that has a frequency of 5 reads. In this case, it's better to cut away this sample because it could introduce false positive or false negative to our analysis. It is important that the frequency of the samples is more or less similar. This is the feature table visualization, both in TXT and in Excel file. We have the ASB-ID, that each ID represents a sequence, like I said before, with at least one nucleotide change. And we have here the name of our samples. For now, we put aside the table and we go to do the phylogenetic analysis. The phylogenetic analysis in QIIME2 are performed with MAFT and FAST3. MAFT handles the multiple sequence alignment and FAST3 generates the tree. We do the phylogenetic analysis because it's necessary to do the alpha and the beta diversity analysis. This index permit to have a quantitative measure and a qualitative measure of the community richness and the dissimilarity respectively. Here there is a little image with plants that explain how alpha and beta diversity work. In particular, in the first plant A, we have 3 bacterial type, and in plant B we have 4 bacterial type. So plant A has an alpha diversity of 3 and plant B has an alpha diversity of 4. When we have to calculate beta diversity, we should compare the 2 plants. So we have 3 species in the plant A, which 2 of them are in common with plant B. So we do 3 minus 2, plus we have 4 species in plant B, but 2 are in common with plant A, so 4 minus 2. We have, as a result, 4. So the beta diversity here is 4. We have, as you can see from the slide, several index for alpha and beta diversity. Each of them measure a different thing. The most used index is the Shannon index. It's a quantitative measure of community richness. So it says to us how many bacteria there are in that sample, in that group, in group analysis. And it's important to compare 2 groups for Shannon index because usually higher is the Shannon index, higher is the number of bacteria, better is the health of the plant, the person. It depends on the analysis. The fate phylogenetic diversity, that is a qualitative measure of community richness based on the phylogenetic relationship between bacteria. The evenness instead describes the uniformity of species abundance inside the community, and the CHA1 indicates the richness, the species richness, so how many species are present in a sample. The beta diversity instead, as we said, measure the dissimilarity within a community. The Jacquard in a qualitative way, the Brechartis in a quantitative way. The Unifract instead uses a phylogenetic relationship, the unweighted one in a qualitative manner and the weighted one in a quantitative manner. QIIME2 gives to us already some plots of the Shannon index. The Shannon, the alpha and beta diversity in general is calculated with the QIIME diversity command. Use core metric phylogenetic using the phylogenetic tree that we have created before and the table. And we will have as output the core metric result folder that we have inside 1 vector for each index. Here we see the Shannon one because we are we are talking about Shannon diversity, but it will be one for heaviness, one for fate, and so on. And trying to give us this plot, but without the p-value. So we don't know if CRC here have a significantly different alpha diversity Shannon entropy compared to obesis here. But we can do this downstream analysis with R. The same thing for Jacquard distance, where we start from the distance matrix that was created with the previous command. And we will have the Jacquard subject group significance and also these plot that are visualized in QIIME view with the emperor. Each sample is as a different color. Here, for example, the red are the CRC patient, the blue, the yellow, and so on are the obese and the control. And we will see that CRC patients are very dissimilar from other ones. In fact, from the Permanova result, we can see that, for example, the p-value between CRC and obese is significant. Here it's 0.001. Now let's go to the best part of 16S analysis, so the classification of the taxonomic classification of 16S sequence. This can be done with the command feature classifier of CINI, and we can use one of the currently available classifier that CINI gives to us, and we have green genes that is the most used for human, Silva that is the most used for plants, and it can be also used for 18S, but as little problem, so it is difficult with Silva to have the species taxonomic classification. Most of the time we are only able to arrive to genera. After that we have the Unite that is used for the ITS, so for the fungi, and the OMD that is used for saliva in 16S, only for saliva. Alternative, it's possible to create our own classifier. It's very time consuming, but if you need it, you can do it. From the classification, we will have as output the taxonomic QZA, and we can transform it in a TXT and visualize it. We will have 3 columns. The first one, the feature ID, that contains the names associated with each read. If you can imagine that these names are the same that we have already seen in feature table as ASBED. After that, we have the taxon associated to this feature ID. In particular, in the first row, we have the Enterobacteriaceae. And we have the confidence score, that in this case is 99.8%. That is the probability that the feature ID and the taxon association is correct. CINI permits to us also to visualize our taxonomy file, transforming it to QZV using CINI view for sure. And we can choose the level. I choose here the level 7, that is the species level, but you can choose also level 6, that is genera, level 2, that is phyla, whatever you want, and you will see which bacteria and in which percentage are present in each sample. But it's very confusing to see it like this, and we will need for our analysis to analyze a group of sample, for example, the CRC group or the obese group, and we can do it with R. The downstream analysis in R can be done using the CINI2R package, because this package permits to read directly the QZA artifact of CINI2. Here, for example, I read the Shannon vector. Reading the Shannon vector, I am able, with the package ggpub, to plot my results and to calculate a p-value, doing a ANOVA or a Cluster-Wallis analysis. And I will have this plot here with the box, one for each group, and with the p-value. In this case, they are not significant. And I put here all the the alpha diversity index, we can see that the Shannon entropy is not significant. It's more or less the same between the three groups. But if the Shannon entropy is not significant, it doesn't mean that other indexes are not significant too. For example, we can see here that the fat phylogenetic diversity is very different comparing CRC with obesis and CRC with CTRL, so the control. This means that the bacteria inside each group are very different at phylogenetic level. We can also plot beta diversity. Here an example with patients that are positive or negative to HPV-16, and it is possible to create an helix around the sample. You can see also here that depending on beta diversity index, the helix plotted are very different. And you can calculate the p-value, as I said before, with QIIME2, but also on R, using the PhyloSec package and the vegan package. You can change the method here. Here I put Jacquard only for example. And you can calculate the Jacquard distance matrix, and after that, using the Adonis2 function to calculate the p-value. That is, in this case, is very significant. So you can already see it from the image, but HPV-16 plus HPV-16 negative sample are very different between them. After that, we can also calculate the differential abundance population. It can be calculated with two packages, the SEC2, that is already used for RNA-seq. So if you can use it for RNA-seq, you can use it also for microbiome analysis. And the Ancom BC2 tool that is specific for QIIME2. To compare the abundance between groups, you need to import the microbiome data. In this case, you can do it with the PhyloSec R package. You can import the O2 table, or the SB table, it depends. And you can decide which group compare and do the comparison. Here in this plot, you can see two comparisons, obesis versus control, and CRC, so colorectal cancer patient versus control. Let's focus on this, the pink one. In this plot, we can see that we have a log-fold change and the asterisks. The asterisks represent the adjusted p-value. More asterisks means more significant comparison. If the log-fold change increase, so go to the right, we have that the bacteria in CRC increase compared to control. If it decrease, it means that bacteria in CRC, that is the group 1, decrease compared to the group 2, the control. Here a practical example. We have Streptococcus. This is a plot done in genus level, but it can be done again class level, phylum level, species level, you can choose. And we can see that the Streptococcus increase in CRC compared to control with a very, very significant adjusted p-value. 4 asterisks stand for less than 0.0001. And the level of increase is more or less 5. Roseburia instead, it's in the left part, so it means that it decreases in CRC compared to control ones, and it decreases more or less than 8 times. So CRC patient will have 8 times less roseburia than control patient in a significant manner. And this is the way to read this plot. But we can do another analysis with PICRAS2, as I said before. PICRAS2 predict the functional potential of our microbiome community. Why? Because it bridges 16S amplicon data with KEGG and enzyme collection, and it has circa 20,000 full-length 16S genomes inside it. What does it done? It knows that roseburia produce like the metabolite A, the metabolite B, the metabolite C, and so on. And it knows it for each bacteria, and it calculates from the abundance of each bacteria the abundance of the metabolite produced by this bacteria. And after that, with R, we can do again a differential abundance analysis. For here in the example, the CRC versus control one, and we can understand between the two groups which metabolite is increased or decreased. In this example, we can see that we go to the left with the log fold change. So we had a decrease in the first group CRC compared to the second group, the control. And we have, for example, a decrease in the sorbitol mannitol transport system substrate binding protein. And the dimension of the dot here depends on the adjusted p-value. This is only a way to plot this analysis. You can also plot it with bar plot like we have done for differential abundance analysis. But here is interesting that you can also put color for groups. For example, the green group is the environmental information process. The yellow group is cellular processes. And you can have information not only on which bacteria you have in your group, but also of which pathway, which metabolite they change. That's it. Thank you for watching us today. If you have any question, this is the right time. Thank you.